Hello and welcome back to the Biochemistry for Health Sciences channel. In the next few videos, we shall talk about how cells divide and related topics such as nucleotide synthesis that's required for cells to divide, DNA synthesis or DNA replication that is also required for cells to divide, as well as apoptosis. In today's video, we shall focus on labile, stable, as well as non-dividing cells. So we all started off as a zygote, basically the egg that was fertilized, and the zygote is about 1 milligram, less than a milligram. So imagine that zygote grew into an adult. Let's say an average adult is 70 kilograms, it's about 154 pounds. So we are talking about a 70 million times increase in weight as the zygote becomes an adult. So this increase in weight happened because there was an increase in cell number. That is, the cell divided into 2 cells, 4 cells, 8 cells, and so on. And then there's also an increase in the size of the cell. So clearly cell division is a very important thing as the embryo develops into an adult. And as we shall see later, cell division is also important in an adult as well, because we do have a lot of cells, especially in our bone marrow, that divide very, very rapidly. A few definitions about stem cells. So we have totipotent stem cells. They appear very early on, for example, in the zygote and the morula. So these are cells here, the zygote as well as the morula, are rich in totipotent stem cells, and these cells can differentiate into any cell type, including the extra embryonic tissue such as the placenta as well as the amnion. Remember, these tissues are discarded after birth. The totipotent stem cells then will differentiate into pluripotent stem cells that are predominant in the blastocyst. And these cells can differentiate into any cell type, but not the extra embryonic tissue such as the placenta and the amnion. And then the pluripotent stem cells can differentiate into multipotent stem cells. These multipotent stem cells are also found in the adult. And in the adult, we call them adult stem cells or somatic stem cells. Now, multipotent stem cells can differentiate only into limited number of cell types within a tissue or cell lineage. And examples of multipotent stem cells are the hematopoietic stem cells, which differentiate into RBCs, white blood cells, as well as platelets. We have the mesenchymal stem cells that can differentiate into cells of the bone, cartilage, fat, or muscle. We have the epithelial stem cells that can differentiate into epithelial, such as the epithelium of the skin, as well as the epithelial lining of the GI system. And then we have the neural stem cells that can differentiate into neurons, astrocytes, and oligodendrocytes. So what happens as the stem cells differentiate? Now, the amount of DNA and the type of DNA is the same in every cell. So in terms of the biochemical aspect, when we say a cell differentiates, what we really mean is that access to certain genes, remember all the cells have the same DNA, it's just that the access to certain genes in the DNA has become restricted as the cell differentiates. So during differentiation, some of the genes become silenced. So certain genes are what we call silenced, or they are turned off. And then some genes, as the cells differentiate, they become activated or turned on. And when these genes are turned on, that will allow the transcription and then the subsequent formation of proteins in that cell. And recall that the adult has about 200 different types of differentiated cells or specialized cells, and these cells really differ in the amount and the quality as well as the quantity of the protein that is present in the cell, because as we know and we appreciate by now, proteins basically carry out everything for cells. So a red blood cell has a different protein profile than a certain neuron, and the neuron will have a different protein profile than the hepatocyte, and so on. And that's what makes these cells specialized to carry out specialized functions. Now, gene silencing as well as gene activation is actually driven by epigenetic mechanisms. And examples of this include DNA methylation. So by methylating the nucleotides in the genes, in the DNA, one can inhibit access to the genes. Histone modification, chemical modification of histone proteins. Remember, histone proteins wraps the DNA, and that can make the gene either more or less accessible for the transcription machinery. Another epigenetic mechanism is chromatin remodeling. So chromatin structure can be rearranged by chromatin remodeling proteins, and this will allow the cell to control which genes get access and which genes will not be accessed by proteins. One importance of understanding cell differentiation is in this area of regenerative medicine, where we are trying to find therapies that will help us either replace, repair, or regenerate damaged or diseased cells, damaged tissues, or damaged organs. Regenerative medicine involves gene therapy, where genes can be modified, stem cell therapy, where we can introduce stem cells into the body to replace lost cells, as well as tissue engineering, where we can make new tissues or organs to replace the disease or non-functioning tissues or organs. So as we develop from an embryo, as mentioned earlier, the totipotent cells will become pluripotent cells, and then these pluripotent stem cells will then become multipotent adult stem cells. And then these multipotent stem cells will differentiate into the various types of specialized cells, the 200 or so different types of specialized cells. Now, the size of any given cell population in our tissue or organ really depends on the rate of cell division, as well as the rate of cell differentiation and the rate of cell death. Now, normally cell death happens by what we call programmed cell death or apoptosis. So depending on these three factors, that will determine the size of any given cell population in our body. Now, the size of the cell population will obviously increase if cell division increases and or if cell death decreases. Now, abnormal states that lead to increased cell proliferation include neoplasia or cancer, and this is a topic very closely related to cell division, and we shall cover this topic in a future video. So based on their proliferative capability, the cells in our body in an adult may be divided into three types. So the first is what we call labile, or in other words, continuously dividing cells. So these are cells that can divide very rapidly. Then we have the second category, which is stable or quiescent cells, which are cells that normally do not divide, but when stimulated, they can be made to divide. And then on the other extreme, we have cells that do not divide at all, and we call them non-dividing or permanent cells. So let's look at each one of these. Labile or continuously dividing cells, as the name suggests, these cells continue to divide throughout life, replacing those that are destroyed. Examples of these cells include the epithelial cells of the skin, the vagina, the cervix, uterus, oral cavity, the GI tract, urinary tract, as well as cells of the bone marrow. So for example, the bone marrow contains hematopoietic stem cells, or HSCs, that will generate all the white blood cells as well as red blood cells and platelets. And their generation capability is tremendous. For example, if you look at RBCs, we produce about 2 to 3 million RBCs per second. That's very, very rapid cell division. In the intestine, the adult stem cells, there which are located near the base of the crypt, they divide quite rapidly. It takes about 4 days to regenerate the whole GI lining. And then in the skin, we have adult stem cells that are found in the lower layer of the epidermis. We have stem cells found in hair follicles as well as sebaceous glands. In these, skin replacement or replenishment takes about 1 to 2 months. So these are examples of what we call cells that continuously divide throughout our life. We call them labile or continuously dividing cells. And then we have these stable or quiescent cells. Now these cells, such as hepatocytes, the renal tubular cells in the kidney, smooth muscle cells, satellite cells near the muscle cells, as well as the endothelial cells, they normally have a very low level of cell division. Okay, so they don't divide much at all. But under appropriate stimuli by signals, what we call mitogens, examples of mitogens are certain growth factors. So under appropriate stimuli, these cells can undergo rapid cell division, and they can actually replace the original tissue that is damaged or destroyed. One dramatic example of this is the liver, which we'll talk about under cell division. Even if 60% of the liver is destroyed or removed, for example, during certain surgeries, we can replace this lost tissue. Another example is the rapid proliferation of fibroblasts that occur in wound healing. Okay, so basically these cells, they don't divide much at all, but when they are stimulated, they can rapidly divide. And then on the other extreme, we have cells, non-dividing or permanent cells. These cells have basically left the cell cycle and cannot divide after the postnatal life. Examples of these cells are neurons, matured skeletal muscle cells, not the satellite cells that exist between the basal lamina and the muscle cell plasma membrane, as well as cardiac muscle cells. So neurons, the matured skeletal muscle cells, and cardiac muscle cells are examples of non-dividing or permanent cells. So for example, the heart muscle is an example of a non-dividing or permanent cell. And this means that if there is an injury to the heart muscle, this is what we see in a myocardial infarction or heart attack, what happens is that damaged tissue is simply replaced by connective tissue, by scar tissue instead of the original muscle tissue. And that will lead into the loss of function. And that loss of function in this case will lead to impaired cardiac function. A few words about this very exciting research here. This is the production of induced pluripotent stem cells. So in this technology, differentiated adult cells can be induced to become pluripotent cells. Okay, so these differentiated cells in an adult can actually be made to become pluripotent stem cells. And this is done, this conversion of these differentiated cells back into stem cells. Remember, normally we always go stem cells to differentiated cells. But here we are going from differentiated cells back to stem cells. This can be done by introducing what we call pluripotency-associated genes or reprogramming factors into these differentiated cells such as fibroblasts. So when these reprogramming factors are introduced into a specialized cell or differentiated cell such as a fibroblast, the fibroblast becomes a pluripotent stem cell. Four important reprogramming factors have been found. We call these the Yamanaka factors. These include the OCT4, SOX2, KLF4, as well as c-MYC. And this technology is a very interesting solution, very important solution that will help us overcome the ethical concerns with the use of embryonic stem cells in regenerative medicine. Okay, so this is called induced pluripotent stem cells, iPSCs. In the next video, we shall look at cell division. So until the next video, thank you for supporting this channel and have a wonderful day, and we'll see you soon.